All right, ladies and gents, welcome back. We're going to pick up with our last topic for protein synthesis, and that's just talking about how cells can sort of regulate this idea of gene expression. So when we talk about gene expression, we're talking how do your cells take the information from the DNA, go through transcription and translation to ultimately produce those proteins. And if you think about it, these gene expression processes are very much energy and resource intensive. They need all of this ATP in order to build these molecules, even though those enzymes are there to help lower that energy cost. It's not free. We need energy to build the enzymes in the first place. So your cells, whether they're prokaryotic or eukaryotic, have to be able to alter that gene expression in response to changing environments. Think about it. This is sort of favored by natural selection. Why am I going to produce a protein that my cells don't need at the moment? And so the way in which prokaryotes versus eukaryotes alter this gene expression does look a little different. When we're talking about multicellular eukaryotes, most of the alteration in gene expression isn't so much to their external environment, but more towards their internal gene expression regulating their development and being responsible for how we get all of those different cell types in your body. Because if you think about it, you've started off as a single cell full of 46 chromosomes. Every time we go through mitosis, I'm making another cell with 46 chromosomes worth of information. And yet, every cell in your body doesn't access all 46 chromosomes worth of that information, and it shouldn't, because I'm only wanting to access the information that I need in order to do my job. So the subset of your genome, the subset of your DNA responsible for telling me how to be a heart cell, should be what I'm accessing if I'm a heart cell. If I'm a neuron, I need to access another set of information. And so we've got things in place to alter that gene expression in order to ensure that the cells are doing their job and not trying to do the job of somebody else. From a bacterial standpoint, from a prokaryotic standpoint, I'm a single-celled organism, so I need to access my genome because I need to do everything in one cell. But I don't necessarily need to express every gene at every moment of my life. And natural selection clearly is going to favor the bacteria that over time had sort of figured out, and I put figured out in quotation marks because there is no sort of brain process to natural selection, but things occurred to them over time where they ended up being able to regulate which genes and which proteins they were producing at different times. And as a result, that particular variation became more and more common in the population over time because it was more efficient, only produced the products that they needed when they needed, and so they weren't wasting energy and resources producing things they didn't. So let's take a look at what that sort of gene regulation might look like in bacterial cells. And bacterial cells have their genetic structure set up in these operon systems. And what an operon system is really is go back to that gene structure, right? Promoter, gene coding, terminator sequence. But within that promoter, I've also got this operator. Think of it like a switch. I could turn it on or I can turn it off. And we do that molecularly. There's got to be a molecule in place. So this particular system is for the lac operon, lac being short for lactose. All right. So if I'm a bacterial cell who has no lactose, no milk sugar in my environment, why am I producing a bunch of enzymes to break down that lactose if it's not there? So they have evolved a system to be able to turn off access to that set of genes that produces the enzymes that break down lactose when there is no lactose in their environment. So here's my RNA polymerase. Remember, that's what binds to that promoter, opens up the DNA so that I can go through transcription and make that mRNA from the genes. But In the case of a lac operon, this system, this gene, generally is turned off. So long as no lactose is there, I've got this molecule called a repressor. Sort of think of it like the off switch. It's cut the cord. It's not allowing anything to gain access. So long as that repressor is there, although RNA polymerase can still bind to that promoter, it can't get any further. It is literally a physical barrier blocking access to this information. But should that bacterial cell end up with lactose in its environment, take it into its cell, that lactose can make its way into its genetic information, and actually it covalently bonds to that repressor molecule. When lactose is around, bonds to that repressor, this guy falls off, and now there's nothing physically blocking. So RNA polymerase comes in, binds to that promoter, no longer has that stop block in the way, and now it's able to access and do its job, open up the DNA, go through transcription, make that messenger RNA, which then can ultimately go and be translated into those enzymes. Should that lactose then disappear from the environment, it no longer is attaching to the repressor goes back on the operator, and then the blockage happens again.
Hi, it's Mr. Anderson, and in this video I'm going to talk about the operon. The operon was discovered in the 1960s by three French scientists, Jacob Louaf and Monod. That's really bad French. But they got to coin the term operon, and basically it means to operate. And so it's mostly found in prokaryotic cells, mostly found in bacteria. There's a few eukaryotic operons, but in general it's just going to be found in bacteria. And so it led to other terms that you've maybe never heard of before, like regulons and modulons and stimulons. Stimulons are groups of operons operating together from one stimuli. And so it just means to operate. That's what an operon means. And so the one that they discovered was the lac operon. They were studying this in E. coli. And so the lac operon is designed for E. coli to break down lactose. So lactose is going to be a disaccharide. And remember, if you're a bacteria living in our gut, you're eating whatever we're eating. And so as the material moves down through our digestive system, they have to be ready to quickly shift depending on what the food is. If it's sugar, they have to be able to break down the sugar. If it's proteins, they have to be able to break it down into amino acids. And if it's a disaccharide, in this case, they have to move it into the cell and then break it down and chemically digest it. And so they use a lac operon to do that. Now, this is a picture of Prague, but that's not how you spell Prague. But it is the way that I remember the parts of an operon. You've got a promoter, a repressor, an operator, and then genes. And so if you can remember Prague, it's going to get you half of the way there to remembering how an operon works. And so the neat thing about bacteria is that if they have a number of different genes that are used to achieve a certain task, they'll put them right next to each other. And so as the RNA polymerase moves down the DNA and makes all of the messenger RNA and makes all the proteins, you're just ready to do whatever the job is. And so the genes are going to be located right next to each other, in this case, in the lac operon, to deal with lactose. What else do we have? Well, we've got the promoter. The promoter is going to be the region of the DNA where the RNA polymerase can grab onto the DNA. We also have an operator, and so think of that like a on-off switch that's going to turn on or off the operon. And then the last thing we have is a repressor. And so this is a lac operon. It's PROG. It's promoter, repressor, operator, and then all of the genes. And so what are we trying to break down? We're trying to break down lactose or deal with lactose. Remember, lactose looks like this. It's a disaccharide. But in this little model right here, we're going to represent it as a little pentagon, a yellow little object that looks. And so all of a sudden, a lot of lactose is present. So what is the lac operon going to do? Well, it's going to deal with that lactose. And so all of this lactose is moving around, remember, but eventually one of the molecules is going to bump into the repressor. As it bumps into the repressor, it's going to change the shape of that repressor. And so a quick digression, everything promoter, operator, and genes are DNA. So they're going to be part of the DNA of the bacteria. But the repressor is a little bit different. It's a protein. It's a protein that came from a different part of the DNA. There's going to be a regulatory sequence, either upstream or downstream of the operon, that creates the repressor. It plugs in nicely to the operator, but once the lactose fits inside it, It's going to change the shape of that repressor. What does that do? Well, it opens up a region where that promoter can allow the RNA polymerase to get on, and then that RNA polymerase is just going to drive right down the DNA. It's going to make a bunch of RNA. It's going to make a bunch of those proteins. What are the proteins designed to deal with? Lactose. And so they're going to break down the lactose. Now all the lactose is gone. What's going to happen to that repressor? It's going to return to its original shape, and that shape is going to fit into the operator. And so if there's no lactose present, we would say the operator is in the off position because the repressor is activated. It's activated. It's sitting inside the operator. It's physically breaking the movement of that RNA polymerase. The RNA polymerase can't move down and code for those genes because the repressor is sitting in its way. What happens next? Well, lactose shows up again. Lactose would move into the repressor, free it up, and then it's going to be able to make those genes. And I've got a simulation at the end of this video that shows you in a little more detail how that takes place. Before we get there, I want to talk about the trip operon. The trip operon does essentially the opposite of that. So the trip operon is evolved in bacteria to deal with tryptophan, or more specifically to deal with the absence of tryptophan. So tryptophan is going to be an amino acid, and it's required to make proteins. It's one of those 20 essential amino acids. And so the trip operon basically is designed to make tryptophan if it's not present. And so we're going to get tryptophan in poultry, in milk for that matter. There's going to be high levels of tryptophan. But if a bacteria doesn't have tryptophan, there's a number of different genes that are required to make it. And so they can make their own. And so we've got an operon called the trip operon. How does that work? Well, again, we've got prog. We've got our promoter, our repressor, our operator, and then our genes. But if we have tryptophan present, so tryptophan is going to be these yellow hexagons. If we have tryptophan is going to fit inside the repressor, and it's going to change its shape so that it fits in the operator. And so in this case, if we have a bunch of tryptophan present, then we don't want to make tryptophan. And so the repressor is going to set that operator to the off position. What happens if all of a sudden the tryptophan goes away? If there's no tryptophan in the diet, well, the bacteria is not out of luck because it changes now the shape of that repressor. And so once it's changed the shape of the repressor, RNA polymerase can grab on, drive down that operon, make all of those genes, and those genes can be used to create more tryptophan. And so it's a great kind of a feedback loop. So in a lacopteron, we're going to turn it on if we want to break down lactose. In a trip operon, what we're going to do is create tryptophan if we don't have it or if it's not present. And I really didn't understand how operons worked until I stopped thinking like a scientist and really started thinking like an engineer. We've got a problem that we have to solve, and one of the great ways we can do that is using an operon. And so I'm going to show you a quick simulation of how this works. It's a PHET simulation. Here's a website. I'll put a link to the simulation down in the video description down below. But it's a great way when I've used it with my students for them to wrap their head around how an operon works. And so we're looking at a lac operon. And to make it simple, we're only dealing with one gene, one of those three genes that are found. And so if we're to look at the simulation, you could try to figure out what everything is, but it's easier if I show you the legend and show you what everything is. And so we've got RNA polymerase kind of bouncing around here. Again, they'd be moving around. There's molecular motion. They'd just be randomly bouncing around. But this is one of the players. We've got the RNA polymerase right here. We could look down below, and I could ask you, now, where is the operon? Is this the operon here, or is this the operon? Well, the right answer is this. This is going to be the operon here because it's got a promoter. So let's put that promoter out here. It's got an operator, an on-off switch, and then it's going to have a gene, in this case the LAC-Z gene. So what's this up here? Well, this is not an operon, but this is going to be another what we call a regulatory sequence. And so this one down here is going to make an enzyme that's going to break down lactose. But what's this RNA polymerase up here going to make? Well, it's coding, it's making a little bit of messenger RNA, but what it's eventually going to make is it's going to make a repressor. Okay, so now we've got the fourth part of our operator. We've got the promoter right here, we've got the repressor, we've got the operator, and now we got the gene. But you can see that the RNA polymerase can't make that gene because the repressor is blocking its way. And so it's in an off position at this point. And it should be. In other words, we shouldn't start making those genes until lactose is present. And so let's add a bunch of lactose. So I'm going to add a bunch of lactose here, and let's watch what happens. Well, if we add a bunch of lactose, and we could speed up the simulation a little bit, what's going to happen is the repressor, and I could pause it right here, that repressor is now having the lactose bind to it, and it's changing its shape or it's changing its conformation. Once the lactose, and you can see it's happened in two cases, once the lactose binds to the repressor, it can't bind to the operator anymore. And so if we play the video, what's going to happen? Well, now RNA polymerase can drive down the operator, down the operon. It can produce a protein. In this case, that's going to be a LAC-Z protein. What's it going to do? It's going to digest and break down all of the lactose that's present. And you can see that it's gobbling up all of those lactose. The only lactose left is those that are bound to the repressor itself. What happens eventually, all of the lactose is going to disappear. In other words, we will have broken it down. What happens now? Well, that repressor is going to bind to the operator again, and now we can't make that protein to break it down. And so it's a wonderful feedback loop. It's a feedback loop that deals with a bunch of lactose present. Because E. coli would be silly to make all of the proteins to break it down until we've got a bunch of lactose present. And so that's an operon. What is it again? It's all of the genes and then a way to control those genes in one tidy little package. And I hope that was helpful.
Now, I mentioned to you in eukaryotic cells, some of that is also occurring, but we do it in a slightly different way. We're going to block transcription from happening in our DNA, but it's usually by adding things to either the DNA itself, the actual nitrogenous bases, or by adding things to those histones. So if you remember back when we talked about your chromosome structure, right? You've got your double helix DNA wrapped around these protein balls, these histones. And those histones actually have these little tails that stick off of them. And that's where we're able to attach certain other chemical groups. So if I add something like a methyl group to that histone tail, I can make them get extra sticky and make them all stick together. I can also add things like acetyl groups, so another functional group, and instead of getting them to be all sticky, they fall apart. So these acetyl groups, these methyl groups, all of these functional groups that can exist and can attach to different places in your DNA allow us to go into a particular gene and either open it up. Remember, chromatin is loose DNA where I now have access to information, or I can condense it, make it all stick together, more like that supercoiled chromosome structure, and now I don't have access. So loose, accessible, condensed, not accessible. So when we went through mitosis and meiosis, we took the entire chromosome and condensed it. I wasn't accessing the information at that point because all I wanted to do was move it and move it safely without breaking it. When we talk about gene regulation here in this case, now I may be just doing it to certain portions of the chromosome. So rather than taking the entire chromosome and condensing it, I'm taking maybe 2/3 of it and condensing it and leaving the other third accessible for that information. And this is kind of a really cool way that your cells do this. It's what we talk about as epigenetics. So it's sort of epi above your actual A's, T's, C's, and G's. And there's some really interesting things that come about the way that this works. So when we talk about that kind of idea of nature versus nurture, how much of the way that you function, the way that you look, has to do with your actual A's, T's, C's, and G's, your actual DNA sequence, as opposed to how much of it has to do with environmental factors, things that you're exposed to in your everyday life. And we always knew that there was some combination of both. You certainly can't alter your DNA sequence. That's something that you inherit from your parents, and there's no way to change it. But your epigenetics and these functional tags seem to be a little bit more flexible and a little bit more influenced by your environment, at least in certain situations anyway. So inheritance of these functional groups can come directly from your parents. All right, so what they do can influence this, but a lot of it actually gets washed away when that baby is created. And so things that you do over your lifetime can change the way that these groups attach. So take a look at the video that's coming up next, and I'll let them sort of explain a little bit more in detail, but it's some really kind of cool stuff that's out there in the research at this point.
So say you have a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy, but it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now, I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, Clone probably has a tattoo on its face. And if Clone had stayed in the circus, Clone probably, professionally rides a unicycle over a tightrope, so Clone is probably in pretty good shape. But then again, Clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic Tater Tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So, at 50, you and Clone would probably look pretty different from one another. But it turns out, you don't just look different, you actually have become different. Now, if some scientists looked at your genetic code, they would say that your DNA was still exactly the same as that other person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference, and if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely, potentially changing the message of that paragraph. The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern, and that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life. But these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic information in a cell isn't permanent. It can change throughout your life, and it can be hereditary. And it can change over time, especially when your body is going through a lot of changes, like, say, during puberty. A bunch of methyl groups kick in and they're like, okay, so you guys over here, you're gonna have to start growing hair, and you guys over here, I really need you guys to get behind giving this guy some really horrible acne. Or like when you get pregnant, which hopefully I won't, but when you do, your epigenome has to be like, all right, guys, everything's about to get a whole lot bigger in here, and we're gonna have to pass something the size of like a Merriam-Webster's dictionary through that little pipe over there, so let's get this thing done. But it's not just these dramatic times when the epigenome is changing. It changes subtly throughout our entire lives, and it changes based on a lot of environmental factors, like what we do, what we eat, what we smoke, and how stressed out we are on a daily basis. Scientists have found that things like a bad diet can actually lead to methyl groups binding to the wrong place and making mistakes. And with those bad instructions, cells become abnormal and become a disease. And then basically all hell breaks loose and you get cancer or something. Epigenetics is a very young science, though we've known about the epigenome since the 1970s. It's only in the last 20 years that we've even known what effect these epigenetic tags are having on our DNA. And even after they got all that business straight, scientists still thought that all of our epigenetic tags were stripped off of our genome before they were passed on to our children. So if you started smoking 10 packs a day when you were 10 years old, that would certainly be a horrible health decision for you, but you wouldn't necessarily be harming your unborn children in any measurable way. However, the thinking on that is changing pretty rapidly, because it's true that a lot, even most of the epigenetic information from a parent is stripped off of the embryo's genome in the first few days, and fresh ones are created specifically for this new person. However, some of these tags get stuck on the genome and are passed down from generation to generation. And it just so happens that the more they study this, the more it looks like bad epigenetic information is being passed from generation to generation. And this is a whole new way to think about how we pass information between generations. Your grandmother was making dietary decisions that affect you today. As we experience all of these new strange epidemics, diabetes, autoimmune disorders, cancers that weren't appearing in previous generations, it's starting to look like these may be caused by epigenetic information passed down from our parents. I know, it's such an unbelievable buzzkill. There is no point in our lives when we can do anything without guilt anymore. The discovery that the environmental factors of parent experiences can be passed down from generation to generation was sparked in the 1980s. And this happened when some scientists were looking at the birth and death records of some people who lived in 19th century Sweden. This is a weird place to find a genetic revolution, but there it is. It wasn't just any place in Sweden. It was in Norrbotten, which is the northernmost county in Sweden, which is literally in the Arctic Circle. And despite the fact that Norrbotten was literally the worst place you could possibly choose to live in Sweden, there were some people living there in the 1800s. And they were completely cut off from the rest of the world. Let me clarify. These people were isolated. Like, if they didn't have a good crop year, people died. No, it wasn't ideal, but, they didn't ask me my opinion, so I'm not giving it. So anyway, these people subsisted entirely on what they grew and the animals that they raised. And like I said, sometimes they starved. But sometimes they had huge, bountiful years of plenty. And when it happened, people totally went ape crackers. I mean, of course they did, because they were so freaking hungry, and then all of a sudden there was all this damn food everywhere. Anyway, there was a public health specialist who was looking at the effects of the people who grew up in the really bad starving periods of time versus the people who grew up in the eat all you can at the smorgasbord years. And you might already be guessing what they found out. People who went from relatively slim pickings to feed your face until you have to barf and then do it all over again in a single season, those people died an average of 6 years sooner than their starved out counterparts. And you know what sucks? So did their kids. And so do their kids' kids. Boo epigenetics. So, of course, now we all know, and we're all going to stop doing unhealthy things starting today. Unless the damage is already done. The damage is almost certainly already done. But hey, epigenetics brings good tidings along with the bad ones. For instance, we now know that certain types of cancer are caused by misplaced epigenetic tags, and scientists are now developing drugs that can silence the bad genes that were supposed to be turned off in the first place. Additionally, until recently, we thought that genes were the end-all and be-all of who you got to be. They were your blueprint, and you couldn't escape them. This outlook is not just kind of depressing, it also leads to a yucky sort of social prejudice. Because when you look at data without considering social and epigenetic factors, it might look like people with less money are less intelligent. So just like 15 years ago, there were scientists saying things in public, mind you, like, some people just have good genes for intelligence, and it just so happens that the poor people don't. Oh, it's so sad. Too bad, poor people. But you have bad genes. Well, it turns out that that is not even a little bit true. Not only are there a huge variety of social factors that affect how well people do on intelligence tests, but a genetic trait is also not just a product of genes, it's also a product of environment. Any one person's genome is determined by any number of decisions made by any number of their ancestors. And right now, you are making decisions that are going to affect people who are alive long after you're dead. No pressure or anything. And so I'm glad you've watched this, because when you and your clone are in your 60s and you sit down, have lunch, and he brings his son and his son has a tattoo on his face, he did not inherit that from you. This is Hank Green for The SciShow. We hope you learned something.